The deadliest virus on Earth doesn't need to shred your brain to kill you. It can hide for weeks, hijack your nerves, and strike only after it's too late. In the 1970s, something strange began falling from the skies over the forests of Western Europe. Helicopters scattered millions of vaccine-laden chicken heads across the wilderness, and foxes and other wildlife stumbled upon these peculiar gifts with confused delight. It was one of the most audacious public health campaigns in history, a bid to vaccinate entire wild animal populations from the air against a virus that had been terrorizing humanity for at least 4,000 years. The campaign eventually succeeded across 24 countries, eliminating fox-mediated rabies from nearly 1.9 million square kilometers of Western and Central Europe. It remains one of the greatest wildlife disease eradication efforts ever attempted. The target was the Lissa virus, better known as rabies, a pathogen so ancient that it appears in writings from 2000 BCE, including Babylonian texts describing the legal liability of owners of rabid dogs. Named after Lissa, the ancient Greek spirit of mad rage and frenzy, it has shadowed our species across every continent except Antarctica. And yet, despite millennia of cohabitation and decades of modern science, rabies remains in many ways a mystery. We still do not fully understand why it kills, which, for the deadliest virus known to medicine, is a remarkable confession of ignorance. What makes the Lissa virus so extraordinary is not its complexity. It is, in fact, almost laughably simple. With only 5 genes encoding 5 proteins, it is among the most stripped-down pathogens in nature. And yet, those 5 proteins allow it to pull off feats of biological engineering that continue to astonish researchers, invading a mammal, vanishing from its immune system, hijacking the nervous system's own machinery, and traveling from a bite wound all the way into the brain, sometimes over the course of an entire year. Everything begins with a bite. Most human cases worldwide involve a dog, and the saliva deposited into the wound carries millions of viral particles. The virus's immediate target is not the bloodstream, it is the nervous system. Specifically, it wants neurons, the long electrochemical wires that carry signals throughout the body, which can stretch up to 1.5 meters in length, with their cellular machinery concentrated at one end and a signaling terminal at the other. The Lissa virus is thought to bind to receptors at the neuromuscular junction, the point where nerve meets muscle, and slip inside nerve cells while their chemical signaling processes are underway. Once inside, the virus faces an immediate logistical crisis. It needs to reach the neuron's cellular nucleus to take over the cell's machinery and begin replicating. But in a cell that can stretch for over a meter, the nucleus might be very far away. Neurons, like all cells, are threaded with microtubules, internal scaffolding that doubles as a transport system. Along these tracks run dynein motors, protein machines built from around 50 different components, 10 times the complexity of the virus itself. Their normal job is to ferry cargo from one end of the neuron to the other. The Lissa virus uses one of its 5 proteins to hijack these motors entirely, commandeering them to carry the viral package toward the nucleus. From there, the virus can replicate and begin its slow, inexorable march toward the brain, jumping from neuron to neuron, advancing sometimes only millimeters per day. This is where rabies diverges sharply from almost every other viral infection, and where its true genius becomes apparent. The human immune system is extraordinarily capable. When most viruses invade cells, those cells raise an alarm by releasing interferons, a family of signaling proteins that do exactly what their name suggests. They interfere with viral replication. Interferons alert immune cells, suppress protein production to starve viruses of building materials, and crucially, They instruct infected cells to display their contents through molecular windows on their surface called MHC class I molecules. These windows allow immune cells to inspect what is happening inside and, if they detect viral components, order the infected cell to self-destruct, eliminating the virus along with it. The lyssavirus blocks all of this. It suppresses interferon production in neurons, rendering itself effectively invisible. It replicates without killing its host cells, which would itself trigger alarms. It creeps slowly from neuron to neuron in a way that generates minimal cellular debris. Research using human stem cell-derived neural cultures has confirmed that lyssavirus infection produces a striking absence of neuronal apoptosis, the kind of cell death that normally signals an immune response. The virus travels through the nervous system like a ghost, generating so little disruption that weeks, months, or in rare cases, even years, can pass before the immune system detects anything is wrong. This incubation period is highly variable and depends on the location of the bite, the amount of virus introduced, and the route to the brain. A bite near the face can produce symptoms in days. A bite on the foot may allow the virus to travel undetected for many months. This is also what makes post-exposure vaccination possible. Because the virus spends so long en route to the brain, there is a window, unique among fatal viral infections, during which a vaccine administered after exposure can still prepare the immune system fast enough to intercept it. Once the virus reaches the brainstem, no known therapy can save the patient. This is stated flatly in the medical literature, and it is not a soft consensus. It is an empirical wall that medicine has not yet been able to breach. A highly publicized treatment protocol developed in 2004, which involved inducing a therapeutic coma along with antiviral drugs, generated enormous hope after a single patient survived. But subsequent attempts have failed in at least 64 documented cases, and researchers now largely agree that the protocol itself is not responsible for the rare survivals, which appear to be related to unusual immune responses the patients mounted independently. Here is the deepest puzzle of the disease. Brain tissue from rabies patients frequently shows minimal damage. There is no mass cell death, no dramatic tissue destruction, yet the patient dies. The current best understanding is that lyssavirus kills not by destroying the brain, but by disrupting the way neurons communicate with each other, so thoroughly scrambling the brain's internal conversations that it simply cannot function anymore. The result is a cascade of neurological symptoms: confusion, aggression, paranoia, the famous and dramatic hydrophobia, a terror of water triggered by painful spasms in the throat when swallowing is attempted, followed by progressive paralysis and organ failure as the patient slips into a coma. The virus, meanwhile, begins its return journey. After traveling for weeks or months toward the brain, it reverses course, a feat that remains mechanistically unexplained even after decades of study, and migrates into the salivary glands. This is how the cycle continues. The infected animal bites, the saliva carries millions of virions, and the virus finds a new host. In global terms, the scale of this cycle is staggering. According to the World Health Organization, rabies kills an estimated 59,000 people annually, though the CDC places that figure closer to 70,000, with significant underreporting suspected in affected regions. Over 95% of those deaths occur in Africa and Asia. Approximately 40% of victims are children under 15. The most disorienting fact about rabies is not how it kills. It is that it does not have to kill anyone. Rabies is, by every measure, 100% preventable. Post-exposure prophylaxis, a series of vaccine doses administered after a bite but before symptoms appear, is virtually 100% effective when delivered promptly. Over 29 million people receive rabies vaccines annually around the world. The modern vaccine, a descendant of the first rabies vaccine developed by Louis Pasteur and Émile Roux in 1885, has saved hundreds of thousands of lives each year and remains one of medicine's most remarkable tools. No other fatal infectious disease offers a post-exposure window during which intervention is still fully curative. And yet, the deaths continue, concentrated almost entirely among the rural poor in low-income countries, where post-exposure treatment costs around $108, an impossible sum for families living on $1 or $2 a day. The WHO, the FAO, and the World Organization for Animal Health launched a global initiative called 0 by 30, aiming to eliminate human deaths from dog-mediated rabies by 2030. Progress has been real. The Americas reduced cases transmitted by dogs by approximately 98% between 1,983 and 2024 through coordinated vaccination campaigns. The oral wildlife vaccination program that began dropping chickenhead baits across European forests in the 1970s eventually eliminated fox-mediated rabies from vast stretches of the continent, with the first field trials in Switzerland in 1978, expanding to 24 countries over subsequent decades. What is required to finish the job is not scientific innovation. The tools already exist. What is required is political will, equitable access to vaccines, and a sustained resistance to the growing cultural suspicion of immunization that threatens to unravel gains made over generations. The Lissa virus has survived for thousands of years by being patient, invisible, and opportunistic. It will exploit every gap in our defenses. The 59,000 people who die from it each year, most of them in places without a functioning healthcare system or affordable access to vaccines, are not victims of a virus we cannot stop. They are victims of the distance between what science knows how to do and what society has chosen to fund. The monster is real. The cure is already in our hands.